Hello everyone present online. Now I'm going to discuss about today's practical water or wastewater disinfection. But what do you understand the differences between disinfection, sterilization and removal of bacteria? Removal of bacteria is just defined as the reduction of pathogenic bacteria due to natural purification and treatment process. And sterilization is just only defined as the functionally inactive of bacteria but not killing the bacteria. But the sole purpose of disinfection is to kill all pathogenic bacteria as well as to prevent the spread of waterborne diseases. The chlorine dosage is used to disinfect the domestic wastewater effluent and it requires contact time of 15 minutes. Which results in a reduction of 99% of coliform content in effluent. What do you mean by chlorine demand? It is just only defined as the difference of amount of chlorine applied and the residual chlorine at the end of contact time. And this chlorine demand also varies with pH, temperature, contact time and amount of chlorine applied. And the sole objective of this experiment is to estimate the chlorine demand and break through chlorine dose for water and wastewater and to remove the COD by chlorination, etc.
The procedure of this experiment involves making the stock solution initially. Stock solution was made by adding 10 gram of bleaching powder in 500 ml water. That's how we made the stock solution. And now different concentration of stock solution will be added in all of them. We have 10 BOD bottles of tap water and 10 BOD of wastewater. What we'll do is we'll add stock solution of varying concentration in all of them. For example, here we are adding we'll take it in the pipette and we'll add it in the BOD bottle. Similarly, we'll do it for all the bottles and we'll keep it after varying various concentrations in the BOD bottles. We kept it in the incubator for 30 minutes. Now after 30 minutes, we'll take out the BOD bottle. We'll take a sample of this and we'll perform residual chlorine and COD experiments on it in the way we did it in the first semester. After that, the readings were noted. For the residual chlorine determination, in the sample, we took 10 ml of sample and then we added potassium iodide crystals and glacial acetic acid. We kept it in the refrigerator for 5 to 10 minutes and we have taken it out. Now what we'll do is, we'll add starch indicator. See, the starch indicator, the color changed. Now we will titrate it with sodium thiosulfate. The initial reading was already taken. We will titrate until it turns colorless. It has turned colorless, and so then we can obtain the results from the volume consumed. For the COD experiment, we take 2.5 ml of sample.
Now we are going to add 3.5 ml espresso pour.
Now I am going to add 1.5 ml of K2CR2O7.
After this, we will add a pinch of mercuric sulfate. And we will close the cap and keep it in the COD digester.
We'll keep the tubes in the digester for 2 hours.
नंबरिंग तो बताना है तो नंबर्स आपने रखे हैं ना.
The numbering has been done. Tap water and 10 samples of that, and wastewater and 10 samples of that.
Machine be working.
We'll switch on the COD digester.
Then go to profile number 19. Zoom in, zoom in, zoom in, zoom in. क्या दिख रहा है आपको? बैठ जाओ. क्या दिख रहा है आपको? हाँ दिख रहा है थोड़ा. लाइट बंद करो. Program, then press enter. So which program? Program number 19. So you have to press on 19, 17, 18, 19. Enter. Profile we don't want to check. It is at 150 for 120 minutes. Enter. Enter. Second video. So it has started. ठीक. Current temperature is 26. Profile number 1. Set we have final is 150. Initial time is 00 and for 120 minutes. So it has not started since temperature is only 27. ठीक. So once it reaches 150, then timer will start for 120 minutes.
Next point of our practical is the environmental significance of the disinfection. The first is to remove the color and the odor of the water. Second is to kill the pathogens. Third is at the start of the main line of the distribution, if you add the sufficient amount of chlorine, then there will be availability of the residual chlorine at the end point of the distribution system, so as it will take care about the excess pathogens which are available or if possible at the end point of the distribution system. Then there is next one environmental significance of disinfection is swimming pool. In swimming pool, you have to determine the residual chlorine. If excess chlorine available in the water of the swimming pool, then it will affect the human health, or it will get the ill effects to the human health. So that's also the environmental significance of this practical. Then in the industrial wastewater or the domestic wastewater, there also the should be determination of the chlorination. Then one more environmental significance of disinfection is it will also helpful in checking the source of the contamination of the wastewater and the source of the leakage in the main pipe. So thank you.